In this video, we are going to look at the biology practicals. So here we have some of the apparatus that we'll be using to do practicals, or some of the apparatus that we'll be using to do experiments. Okay, so the first thing that we are going to look at, the uh, external structures of the eye, the external structures of the eye. Okay, so to view, or to in order to draw the external structures of the eye, we'll be making use of the mirror. Okay, so mirror is one of the apparatus we'll be using to do these practicals. So here we have some of the external structures. So some of the external structures, these are structures found outside the eye. So we can see eyelid, we can see pupil, we can see sclera, we can see iris. So these are some of the structures we'll be looking at. Okay, these are some of the structures we'll be looking at. From there, we are also going to look at the common types of eye defects, the common types of eye defects, or the common types of eye disorders. So the first one, we are going to look at short-sightedness, short-sightedness, also known as myopia. Okay, the second one, we are going to look at long-sightedness, also known as hypermetropia. The third one, we are going to look at distorted vision. Distorted vision is also called astigmatism. The last one, we are also going to look at the age-related long-sightedness, also called presbyopia. So these are eye defects we are going to look at in this video. Let us start with the short-sightedness, also known as myopia. So it causes the distant object to appear blurred. So why distant object appear blurred? So the focal point form in the middle of the eye. So the focal point is supposed to form on the retina, but we can see that the focal point has formed in the middle of the eye. Okay, so we have the normal vision here, and we have short sightedness or myopia. So we can see that on the normal vision, the focal point has formed on the retina. So this is the right place where the focal point must be formed, not here. So because of this, the distant object or distant objects appear blurred. We are also going to look at long-sightedness, also known as hypermetropia. It makes close objects appear blurred. So you might be able to focus clearly, but get tired along the way. So what is causing long-sightedness? So we can see that the focal point has formed beyond the retina, or the focal point has formed outside the confinement of the eye. So the focal point has formed here. So the focal point, like I said, the focal point is supposed to be here. So this will cause short-sightedness. The next defect we are going to look at is distorted vision. Distorted vision occurs when the cornea or lens has a mismatched curve. So we can see mismatched curve here, and we can see the normal curve here. So because of mismatched curve, this person is bound to have distorted vision. We are also going to look at presbyopia. So presbyopia is a long sightedness coming as a result of growing old. So as you are growing old, you are subjected to age related uh, age related long sightedness. Okay, so to appreciate the importance of what we have learned, we are going to answer this question, which is coming from 2015 paper 3. Paper 3 is a practical paper, and the question is question 1b. Question 1b, the question reads: a human eye has a standard length of 3.5 centimeters. So here we are given the standard length. In simple language, we are given the normal length, which is 3.5. B, Roman numeral 1, make a labeled drawing of the eye with the aid of a mirror and show clearly as many visible structure as possible. Okay, so they want us to draw. The we need to draw the eye. Okay, so to draw the eye, we need to make use of the mirror. So you need to use the mirror so that you can view your eye. So by viewing your eye, you'll be able to see the visible structures, the visible external structures. Then you need to label these structures. So here we have the external structures. We can see eyelid, we can see pupil, we can see sclera, we can see iris. So you can include some of the external structures not included here. So we can see that the question is carrying 6 marks. So we have answered, we have provided. So this is our suggested answer. This is our suggested answer. Let us move to the next question, which is the question B, Roman numeral 2. Question B, Roman numeral 2, the question reads: measure and record the length of the drawing. Draw a line on the diagram to show the measured length of the drawing. So they want us to measure the length. This time around, they want us to measure the length of the eye. Okay, so to measure the length of the eye, we are going to make use, so forget about the information here. We are going to make use of our ruler so that we can answer this question effectively. So here there was a mistake. Here we're supposed to have 3. Here we're supposed to have 3.75 centimeters. Okay, so therefore even here we're supposed to have 3.75 centimeters. 3.75 centimeters. Okay, then the next question: calculate the magnification of the drawing. Calculate the magnification of the drawing. So to calculate the magnification of the drawing, we are going to make use of this formula. magnification magnification is equal to actual length, or I mean image length over actual length. So we are going to measure the length of our drawing. We are going to measure the length of our drawing. Okay, so we are going to say magnification is equal to image length over actual length. So we are from saying magnification is equal to, so the length of the drawing we said is 3.75. 3.75. Then the length of the standard length or the normal length of the eye is 3.5 centimeters. So if we say 3.75 divided by 3.5, we are going to get 1.1. So 1.1 is our magnification. 1.1 is our magnification. So when magnification is greater than 1, then you will know that the image is greater than the object. Okay, the image is greater than the object. So here we saw that the length of the image was greater than the object. So that is the reason why we have magnification greater than 1. Magnification greater than 1. So we are also going to look at the soil. Soil is the mixture or composition of organic matters. So organic matters, these are matters or these are materials produced by living things. Then inorganic matters, these are things or materials not produced by living things, and other nutrients that support life of plants and soil organism. So soil is the mixture of organic matters, inorganic matters, and other nutrients that supports life of plants and soil organisms. So there are organisms within the the soil. So we have seen that the soil uh, is a mixture or composition. From there, we are also going to look at the types of soil. Number one, clay soil. Number two, sand soil. Sand soil, silt soil, loam soil. So these are types of soil we are going to discuss. Let us start with the sand soil. So sand soil is as the sand soil has the low fertility, or sand soil has low fertilizer in it. Okay, then clay soil has a relative amount of a relative or a sizable amount of fertilizer or fertility. So the soil is wet and cold during the winter, and the soil get backed dry during the summer. Okay, let us also look at the silt soil. So silt soil is fertile. So silt soil is compacted during the summer, and silt soil uh, has a good water retention. What about the loam soil? Loam soil, we can say loam soil is a combination of sand soil, clay soil, and the silt soil. So loam soil is fertile owing to the fact that it has a good number of organic matters. So these organic matters are decomposed, thereby adding fertilizer to the soil. Okay, thereby adding fertilizer to the soil. We are also going to look at the humus. Humus is the dark organic matter in this. I mean, we are going to look at the causes of soil fertility. So what are causes of the soil fertility? So humus is one of the causes, or humus is one of the factors causing soil fertility. So soil fertility is the uh, i- i- soil fertility shows the how much fertilizer is found in the soil, or how fertile the soil is. So humus is the dark organic matter in the soil that is formed by decomposition of plant and animal matter. It is a kind of soil organic. It is the kind of organic matter. So the humus add um, fertilizer to the soil. Soil texture is also one of the factors that helps the soil to be fertile. Okay, so soil texture affects how well nutrients and water retained in the soil. So detention is the ability to keep or to retain water and the nutrients within the soil. So uh, soil texture helps to achieve retention. So retention helps to keep the nutrients in the soil. Soil cover also adds organic matter to the soil, which leads to improved soil. We are also going to look at the causes of infertility. What are causes of infertility? High acidity levels, or high acid concentration. So high acid concentration on the soil may kill the bacteria. These bacteria help organic matters to decompose, thereby adding fertilizer to the soil. So if there is too much acid within the soil, these organic these organisms will be killed. These organisms play a pivotal role in the process of decomposition, or they help organic matters to decay so that fertilizer can be added to the soil. Okay, poor nutritional composition. So if the amount of nutrients within the soil is too low, then the soil is infertile. Okay, unfavorable soil structure. So unfavorable soil structure also causes infertility. High levels of salinity, too much salt in the soil also causes infertility. So these are soils. These are soils. We have sand soil, we have loam soil, and we have clay soil. We are also going to look at the characteristics of these cells. So we need to understand that sand soil has a good number of spaces or pores within it. So in this case, we are going to look at the porosity. We are going to look at porosity across these three types of soil. So porosity is the number of pores or spaces within the soil. So we can see that we have big spaces, we have big spaces, or we have big pores within the sand soil. So for this reason, sand soil allows water to pass through it, allows water to pass through it. Let us also look at the silt soil. So silt soil has a small pores or small spaces. So silt soil does not allow a huge volume of water to pass through it. Okay, let us also look at the clay soil. So clay soil is compacted, and we can see small spaces left within the particles of the soil. So for this reason, clay soil only allows small amount of water to pass through it. Small amount of water to pass through it. So we can see the evidence here. We have sand soil. So since sand soil has big pores or big spaces within its particles, it has allowed a huge volume of water to be collected here, followed by loam soil, followed by clay soil. So these are characteristic of some soils. We are going to answer this question, which is coming from we are going to look at this question. We are not going to answer this question. Reason being, we don't have the samples. So this one is coming from this one is 2016. This one is 2016. The first question, the question reads: You are provided with the specimen A and B, which are soil samples. A1A, using a hand lens, identify the differences between the two specimens A and B. So we are given two soil samples. So we don't know the soil samples that we are given when this paper was written. The next question, so we don't know if the soil sample that was given is sand soil, clay soil, silt soil, or loam soil. The next question, which is question B, place 100 grams of sample A in a funnel labeled A, fitted with a filter paper and, and placed in a conical flask. Place 10 grams of sample B in funnel labeled B, fitted with a filter paper and placed in a conical flask. Pour 10 cubic centimeters of water into each soil sample in the funnel. Measure and record the amount of water collected in 3 minutes in the table sample and make conclusion. So we have soil samples here. We have soil sample A and B. So they want us to know. They want us to give the volume of water collected after 3 minutes. So since we don't know the types of soil, we are not going to answer this question. We are not going to answer this question. Reason being, we don't know the samples, the samples that were the samples that were given. So for this reason, we are going to end here. Okay, we are going to end here. So if you find this video impactful, uh, react to this video, react to this post, subscribe to my channel,